Thank you very much indeed. I'm also delighted to be here today. Actually in the same room as James. So I was listening to him in stereo and he has to do the same to me now. So I'm going to talk a bit about the latest progress on clinical trials of mRNA therapies in PCD. And two important disclosures for me are Recode and Ethris, both of whom are active in this area. So in primary ciliary dyskinesia, as you all know, the main problem is that the cilia don't beat properly. And what James has really talked about is treatments that he can do to help some of the problems that has caused. So some of the problems with bronchiectasis, with mucus, blocking the airways, with infection, with inflammation. But actually what I'm going to talk about is something that tries to actually look at the cause and the problems with the cilia in the first place. So a step back, which obviously will hopefully mean that some of the other therapies may not in the future be needed. This is a picture of Amelia. I think you might have seen a similar one before, getting a nasal brushing. And hopefully you can see the video on the right, which at the top shows cilia beating normally. And at the bottom, the longer you look at it, the worse the cilial function get. And that, as you know, is the main issue within primary ciliary dyskinesia, in that the cilia aren't beating properly, and hence the mucociliary clearance is absent and mucus gets stuck down in the airways. And it's this that some of these trials will be addressing. As you will have heard earlier, but it's important to go through again. This affects many areas where the cilia align, so in particular the upper and lower respiratory tract, so the lungs with bronchiectasis, chronic sinus problems, ear problems, particularly as a child, and then also problems with a reproductive system as well. I know also that you've had in earlier talks today a discussion of the cilia themselves, but it's really important for this talk, and hence I hope you don't mind if I just go into it in a little bit of detail. The cilia, or these hairs that line your respiratory tracts, these in cross-section have various different components that you can see on the electron microscopy image on the left, but also labeled on the right. And it's this combination that allows them to beat. So the outer dynean arms climb on the microtubules, the circles next to them, and because they're all linked together, that causes this swaying movement. And in primary ciliary dyskinesia, there can be abnormalities in any of the different products that you see within the cross-sectional image there, and they can all cause different types of dysfunction or the inability of the cilia to beat and to move, which then leads to them not being able to move the mucus and provide mucociliary clearance. And that's what it can look like in primary ciliary dyskinesia. In one of the most common types, there's no outer dynean arm. And I don't know how you got the laser pointer. How did you get the laser pointer, James? Is it on Morse? Just 1 second. I don't have the ability, but that's how you can see within some of these electron microscopies where you don't have some of the features or some of the proteins. Now, it's slightly different to other genetic diseases like cystic fibrosis that you may have heard of. In cystic fibrosis, all of the mutations relate to one gene or one protein product. And you can have any mutations, one from the mother and one from the father, which can cause the problem in cystic fibrosis. It is a lot more complex in primary ciliary dyskinesia, in that you can have abnormalities or mutations in any of these different parts of the cilial function. And there are lots of different genes that relate to different protein products. And at the moment, about 70% or 75% of PCD has been defined genetically, i.e. we know the mutation in somebody's genes that causes the problem and the lack of protein product. But different problems can lead to different types of primary ciliary dyskinesia. And that's important when we talk about some of the therapies going forward. Now, the genetics of PCD are predominantly autosomal recessive. Now, that means that you need an abnormal gene from both parents. So here you have the pink being the abnormal PCG gene, the blue being the normal gene. And so normally the situation would be that both of the parents of somebody with PCD would be a carrier, and then there is a 1 in 4 chance of the child getting primary ciliary dyskinesia and a 1 in 4 chance of the siblings also having primary ciliary dyskinesia as well. So that's how it is genetically passed on. So moving on, what we've been talking about is the DNA or the genes. So everybody within their cells has the DNA code, and in PCD, that code has a mutation such that there's an abnormality in the DNA encoding the PCD in patients with PCD. Now, what that does is the genetic code is transcribed to produce something called messenger RNA. And that's important because those are the therapies that I'm going to be talking about. That messenger mRNA is then translated and produces the protein. And it's the protein that causes the different aspects of the cilia that I was talking about. So truly genetic therapies are things that alter the genetic readout. So they alter the genes in people, and that can be lifelong. What we're going to be talking about is the mRNA, which is the transcribed or the messenger product from the genes, from the DNA, and that's something that is temporary. And so that is something that is produced by the cells and then is read out into proteins of the cells. And that's essentially what this therapy does. So what it is going to do is so in patients with PCD, you would have an abnormality in the DNA that causes an abnormality or a lack of mRNA or an abnormal mRNA, which then causes the abnormal protein or the lack of protein in a particular part. What we're going to be doing in this therapy is we're going to be replacing the abnormal mRNA and trying to get that into the cells, into the cilial cells. So the idea is that you put the correct mRNA or the correct messenger product into liposomes that is nebulized, and that is taken up by the cells in the respiratory tract that have the cilia. Once it's taken up, the messenger RNA, the mRNA, then gives the message to the cell to produce the protein, and then that protein, which is abnormal, is then produced. The normal protein is then produced, and then it can build the cilia back to how it should be. So that's the idea of the therapy. And so just going back through it in detail, this is the specific. So this is what's been done by one of the companies, which is Recode, which is what we're trialing at the moment. So this is looking just at a specific mutation in patients that carry the abnormality for the DNAI1 gene. And that's a gene that is essential for outer dynean arm building and for this ciliary beat and ciliary movement. And as I mentioned before, so patients that have a mutation in DNAI1 do not have the protein DNAI1, hence have an abnormality in their outer dynean arm, in their cilia. And what that does is it means that the cilia do not beat. So instead of that, what these patients are going to inhale, they're going to nebulize the mRNA, which will then go into the ciliated cells that have the cilia, and that will make the cells produce the protein, which they can't do normally. That protein will hopefully be able to replace the outer dynean arms on the cilia. The cilia will have full protein functionality. That's what the hope is, and they will start beating again. And so the idea is by inhaling this product, the cells that do not have the right message to produce the cells, to produce the proteins for the cilia, get that and start producing it. So where are we up to with this study? So as I mentioned, at the moment, it's just for DNAI1 mutations. And this has been tested at an ascending dose in healthy volunteers. So there were 5 different cohorts of healthy volunteers. This was predominantly done in New Zealand, and this was looking at safety of the product in human volunteers. And with ascending doses all the way up to 10 milligrams, it was found to be relatively safe. The main issue at 10 milligrams was that patients had some fevers that actually responded well to antipyretics, and there were no other significant safety signals that were found. Following on from that, we have then done a cohort with, again, an ascending dose cohort in patients actually with PCD. So with this DNAi1 mutation, they all had a single dose of this nebulized mRNA encoding correct DNAi1. And these were 9 patients that were either in London or in a phase 1 center in Las Vegas. And again, there was an ascending dose to both 3 milligrams and 5 milligrams. And again, there were no safety signals. Again, some mild fevers being the main issue during those trials. So those so far has just used a single dose, and the main output was a safety signal or a lack of a safety signal. So how can we show, or how can the study be designed to actually show whether or not inhaling this product by patients is going to work? So there's different ways, and what the outputs for clinical trials with this product may be are as follows. Firstly, if we look at the second panel, I've already shown that we can use electron microscopy to show abnormalities in the cross-section of the cilia, so particularly in DNAi1 and missing outer arm. So the hope would be that after patients inhale this product, they will start creating the outer arm again because they've got the product that was missing. So electron microscopy is one way of doing it. The second way of doing it is to show that in DNAi1, so the bottom panel here, this is a typical DNAi1 sample where there's no movement at all. Hopefully, after taking the drug and taking a sample, we will be able to start seeing light microscopy again. The panel on the left that I've missed out so far is immunofluorescence. So this is the ability to actually stain the product. So looking for the product DNAi1 in the cilia when we take the samples and we stain them. And then finally, the other way is to have a look at the function. So with people with PCD, when they take a radio aerosol, where they inhale a radio aerosol, then you can see that at the bottom 2, after 2 hours, it's still in the lung because the mucociliary clearance, the escalator doesn't work. In normals, you get a much better clearance of the radioisotope. And that would be another way of showing whether patients that inhale this product actually have some functional benefit and that it seems to work. So where are we up to? So this is now a phase B study, and there's 2 concurrent studies going on, one in Europe and one at our centre at the Brompton in the UK. This is the Brompton Centre. So here we are going to be looking for all of those 3 initial outputs or clinical efficiency outputs. So we'll be looking at the electron microscopy, we'll be looking at the light microscopy to see if the beating works again, and we'll be looking at immunofluorescence. And these are patients that are now being given not just a single dose of this, but multiple doses. So patients are given these products 3 times a week, up to 12 weeks. We're hopefully going to have 10 adult patients with DNAi1 abnormalities, homozygous. And what's going to happen is each patient, when they are screened, have a bronchoscopy, and that's so we can get a sample of the ciliated cells within the respiratory tract at the beginning of the treatment. And then half of them will have a further bronchoscopy at about 4 or 5 weeks, which we'll see again to get the sample to see if the cells structurally look normal now, the cilia, and if they start beating. And then the other half will have them at the end of treatment. And there's some various other clinical outputs that we'll be looking, but that will be one of the main ones. The other study, which has been going on at the same time, which is in Denmark, Germany, and potentially in the US as well, is the same kind of study, but they will be looking at the outcome measure of the radio isotope, so the mucociliary clearance. So two different studies, very similar makeups, but looking at two different efficacy endpoints that I mentioned before. So where are we? So as I mentioned, the single doses have been very well tolerated in both healthy volunteers and the DNAi-1 PCD patients so far. We're carrying on with the follow-up of these single dose patients, and they all seem fine so far. We have started the multiple dose bronchoscopy study at the Brompton. We have had 2 patients that are having active dosing, and are 1 patient now who has had their second bronchoscopy at the 4 or 5 week mark. The second patient who is 1 or 2 weeks into that. We've also had a third patient who's had their initial bronchoscopy and due to be dosed next week, and 2 others who are ready for the first bronchoscopy either just before or just after Christmas. In Europe, there's been 3 patients who have started dosing, so a similar number of patients, and I think they're a week or 2 behind. And this is again some slides from Recode, and this just shows where we are at the moment. So we are in Q3, I think. Yeah, we're in Q3 of 2020. Depends how you do the Qs. Actually, we're towards the end of 2024, and you can see we're on target with regards to that with a phase 1b 12-weeker study. The hope is that if that looks like it's working, that there would then be a phase 2 study that would go on and would start in 2025 to again hopefully lead to registration and a licensed product if things go well. They're not the only company, as I mentioned before, and the beauty of this idea is that the mRNA can be built for different mutations. So the mutation that's been used at the moment is DNAi-1, but another company, Ethris, is looking at a similar thing for different mutations. So CCDC40, CCD39, and DNAi-5, which is the most common mutation that is available. Some are easier to produce mRNA for than others. DNAH5 in particular is difficult because it's quite a big protein, and they're at different stages of development. Now, what's important, I suppose, is to know the frequency of these different genetic malformations. This is some great recently published data from the ERJ. And if you have a look on the left, the most common is DNAi-5, like I mentioned, with DNAi-1, I think 4 along from the left-hand side. And some of the others that I mentioned, like CCDC40, also one of the more common types. But if this works, the idea is that it could be then rolled out for patients with other genetic mutations. So really important. The difficulty is, however, is that there aren't that many mutations throughout the whole of Europe. I mean, this is actually beyond Europe. I can see Argentina there as well within the group of countries. And if you look at the number of patients with DNAi-1, there's only 74. So actually, this is very difficult to actually get people to take part in these studies because the number of pools of patients that are available is much lower than some of the studies that you've just heard from James. So as he mentioned, the largest bronchiectasis study that was done with brensocatib was over 1,000 patients. And here, there's only 74 patients even that we know of that have this mutation. So trying to perform a study and actually start thinking of ways that you can prove and statistically prove that things work is very, very difficult. And so it's very, very hard to get these studies to move forward. So one of the most important messages here is actually that unlike other studies where you can actually say, oh, that's a really interesting study, let's sort of see what happens. We'll get other people to take part, and if it works, then great, the drug will become available. That really isn't the case in studies where it's specific for your genes or for patients' genes, because actually, if we don't get the people who can participate to participate, the study won't be able to succeed, and potentially a way forward for the future of PCD management, could potentially be lost. So that's one of the messages I really want to get across. This is just some other data from the Ethris platform, again showing similar data. This is in vitro data, so in either patient-derived cultures or in vivo data with zebrafish, just showing the same kinds of things, whereby using the same mRNA technology that I've mentioned in liposomes, they can show in either zebrafish or in cell cultures positive improvements. And the platform for Ethris, they're a little bit behind where things are with Recode, but it's a similar idea, whereby the idea would be that once they get further development of the mRNA product, they would be going for single dosing, multiple dosing, and then a cohort looking for biomarkers or to make sure that it works. So I hope I've been able to explain that well. But what I really want to summarize is that these mRNA therapies really have an extreme potential to change treatment for PCD away from trying to treat the problems that not having cilia moving makes, the damage to the lungs, and really going all the way back to the initial problem, to the lack of a protein, and hence the lack of working cilia. They aim to enable ciliated cells to produce this missing protein. It has the potential to provide bespoke mutation-directed therapy. The preclinical data is encouraging, and the phase 1 studies for proof of principle and safety are ongoing. But the patient numbers are really small, and we really need patient engagement and buy-in to enable this program to succeed. Thank you.